Welcome back. In the previous video, I showed you how to get APRICOT using pip or from Git. And we were going to start working with run example, which should be in our tutorial here. So the first thing to do is to get the run example shell script, which I'm going to just copy. We got the run example. Another is to install bioAPRICOT, which we have already installed. As you can see here, we have already running. Third thing is to download the repository. So this is not completely required if you just want to work with the software, but because we want to work with some data sets, which is in the APRICOT repository, we're going to clone it. If you've already cloned it, keep it in the home directory where you're working. So now we have APRICOT, and we have run example. Sorry, I'm just gonna show you with the basic. So this is the script. Going back in here, running APRICOT or with -h, it shows you different subcommands. You can alternatively use Python if you have it located somewhere locally and not globally. So as we were looking at the shell script, we have different variables. How to call APRICOT, just basically APRICOT, but if you, as I said, if you have locally installed, you can define it already here. Then the analysis path, which we have defined as an APRICOT analysis. So whatever you analyze, your input, output, they're all saved inside this folder. Your APRICOT path, which is basically our Git repository, which we have cloned locally. Inside the Git repository, we have the library in APRICOT lib, but if you're working from the global, then you have it in the user local bin. Then you have root database path, which would be our source file. The source file is everything that we require as a dependent. In general, if you're using a Docker image or if you're installing it, you would have it downloaded. But in case of example, we're gonna download it from the GitHub. So these are the different paths that are defined. So let's just go one by one again. We were right here. Needle, we're gonna need it for the later subcommand. We have Zinodal link. If you want to work with a test data set, you can even define which release of PFAM you're going to work. I have added the most latest one. Then we also work with different flat files, which are downloaded in the source file, which is our root database path, under the folder reference domain, reference DB files. For the domain database, you have CDD for CDD path, Interpro in the Interpro path. If you want to work with a specific species, you can define it here, but for the example, we will not be working with it. I have already listed 2 proteins from Salmonella. These are the UniProt ID. As you see here, positive test because it's a RNA-binding protein which contains RMR RNP, LM-like domain, and then you have second, which is negative test, which is also an RNA-binding protein. However, it uses different domain, which we would not be using as input in our case. So the input 1, we're gonna definitely use the UID, but you can alternatively define gene ID, taxonomy ID, FASTA path if you're working with the real test scenario. Domain keyword, it's our second input beside query protein, and in this case I'm gonna give 3 RNA binding domain family name, RRM, KH, and dead. So why I call the second protein as negative, because if I use RRM, KH, or dead, they will not select this protein as the protein of interest because it will not have any of these domains. So this should basically be considered as not desirable protein for our analysis. This is completely optional. It's a classification keyword. If you're working with large data set, you want to classify them based on specific function, you can define them here. But two things to keep in mind, we are just working with query protein, and as domain keyword we have provided RM and Ctet. So what you're looking at here, these are different modules written in shell, which uses different subcommand of APRICOT. I'm gonna go one by one, so I'm gonna comment all these out. Yeah, I'm gonna just work with the setup analysis folder for the moment, which can be run by sh run example.sh. So it created an analysis folder called APRICOT analysis, which is here. Here. In APRICOT analysis, you have input. Inside input, APRICOT takes the query protein, saves it here, extract different information, saves them there. And then you have output, which are numbered based on different subcommand. We'll go one by one, but they are numbered according to how APRICOT runs it. So the 0 is for predicted domain, because the first step APRICOT does is domain prediction. Second is to compile the domain information. Third is to select domain based on their information, their annotation scoring, additional annotation, and summary. You can find the detailed information if you go down in the documentation or in the tutorial. So down here it tells you where is everything, what these folders mean. So what else do we have? So this is our repository, this is the analysis folder, run example, and the source file. Looking inside the source file, there are two information, domain data and reference DB files. Reference DB files are all the flat files and tools that APRICOT requires. Inside the reference, you have CDD and Interpro, PFAM, gene ontology mapping to both CDD and Interpro data, the domain data, and the domain data. Here we just use it as an exemplary one. Now let's go to the second subcommand. Get a small demo file, so we don't have to really install the entire file system. So it copied. Provide input query. So since we are providing to UniProt ID here as comma separated, we should be able to extract information of that. So it downloads the UniProt ID. It downloads all these information from UniProt. It creates the XML feature file. So now if we again go back to APRICOT input, if we look at query protein, it has a file where the query IDs are saved. Beside query proteins, there are two other folders, mapped query annotation and UniProt reference table. So what you saw from the UniProt, APRICOT downloaded it inside this. We can have a look. Let's just use nano APRICOT analysis input. So here is the protein 1, protein 2, tells you the name, gene name, organism length, and gene ontology. And similarly, you have for so in this folder, APRICOT downloaded the FASTA file, it downloaded the XML file, and it extracted the information from XML as a tabular format. So we can show you that. Query feature table. So it has much more information than just what UniProt has in the table that we downloaded. Alright, so now we can already go to the next step, which is providing domain and class keywords. For the domain keyword, if you remember, we have RRM, KH, dead, so it should take those keywords. So it tells you that the keyword for domain selection, keyword for domain classification. Now we can go to the next command, selecting domain by keywords. It takes a while because it has to go through each entry of the database and select only those that's relevant to our provided keywords. So this information has been saved in source file, domain data, CDD, where it downloaded dead related CDD domain, KH related CDD domain, RRM, and they also compiled it here. Similarly for IntPro, dead related IntPro, RRM related IntPro domain, KH, and all compiled. So we can have a look at it. So it's in the source file, domain data, CDD. Let's check all. So as you see here, it has all these entries that associate with RRM. If you go further down, it should have much more information. What was also there in the domain data are the keywords for domain selection, keyword for domain classification, which basically we provided, and based on the information extracted from CDD and IntPro, everything is saved in this one file, which is called all keyword selected domain data. Now we go to the prediction. So if you're working with the entire file system, APRICOT will predict the domains for each protein, but because we're working with demo file, it will actually have the predicted data, but it will show them. So because it already has the analysis for both proteins by IPR and PPT, it says it exists. In another case where we will run the real test, you will see how does RPS Blast or IntPro work. However, we don't have it installed yet. So let's have a look at that. So it should be an APRICOT analysis, output, in the 0 predicted domains. This is the first step. Inside that you have CDD analysis, and for CDD analysis you have the predicted files. So if you want to have a look, let's copy that. So this is the result. So it shows you all the prediction done in this particular protein, what all different domains were predicted, what are the e-value of it. If you further go down, it shows you the sequence conservation, names, score, identity, and so on. The file for IntPro looks a bit different from that. If we just have a look at the same, it has the protein name. It also tells you how long it is, from where it is extracted, start and stop, and the e-value. We can run the next step. Which is filtering domain analysis. So at this point, what we have is APRICOT that selected domains of interest, and we have our query protein with all the domains in it. So now there will be a filtering step where APRICOT will look for the presence of the selected domains in the query protein. If the query protein consists of domain of interest, it will select it, else it will filter it out. So as you remember, we were looking at this protein, which does not have a RRM, KH, or dead domain. However, this protein has RRM or RNM domain, and therefore it should be our positive, and this protein will be filtered out. So this seemed to have worked. Let's see what APRICOT saved. So we had already had a look at 0 predicted domain. We can now look at 1 compiled domain information. So here is for selected data. So let's see if CDT selected something. Sorry, I should open it in the editor. So it selected one protein which has RRM RBM7 or 11. However, it did not select the other protein, and same would go for integral. We can now, so we have already done that. We can now check if it has something. So we have combined and classified. So we haven't done the classification, so we can look at combined. So it basically has one entry, which is RNA-binding protein HFQ, and it tells you if it's selected by parameters or not, what kind of similarity, bit score, identity, and so on are known for it. We can classify. It's not useful at this point because we don't have a large data set. As I said, if you have large data set and large number of protein of interest, you would want to group them, and the classification helps you to group them and navigate through it. In our case, we don't really need it, but just because we are showing if it works or not, it does the job. Going to the next step, which is summarizing it. And the summary is outside in the fifth. So this is our summary file. We can have a look at it quickly. So it shows you there were 2 query input. Total query input that we could extract from UniProt were 2. Which proteins were mapped with the UniProt data set? How many keywords did we use and how many domains could we extract? So there were 2 keywords used. For the domain of interest for KH we extracted 31 domains, and for RM we extracted 294. Domain prediction summary for CTT, there were total 2 proteins with domains, so both of the proteins had a different number of domain prediction. And here it shows you unique domains, meaning that there are several databases that could identify the same domain, but there are about 18 which are unique. And the last one is that passes through the filter. So out of 19, 9 of them were selected as a true prediction. For second protein, we have 54 total prediction, of which 53 are unique, of which 15 are selected due to the parameter filtering. And here it's just a summary. And now if we want to look at what was filtered by the domain of interest. So it turns out of so many prediction, just this protein had one prediction which actually passed the domain filtering parameter and which is of our interest. Similarly, you can identify here for Interpro. However, unlike CDT, for Interpro, we don't have anything that passes the parameter filter. So this was the short overview of how you can check if the tool is working. You can check